Okay, so this is the third part of the lecture. You've already covered carbon and how versatile of an atom it is and all of the great connections that it can make. We've also already covered functional groups. In that lecture, we talked about 7 different functional groups, and the behavior of a molecule can be predicted by the functional groups attached. So in this one, right before we dive into the 4 macromolecules, or the book says the 3 macromolecules and nucleic acid, there's some vocabulary that we need to just very quickly lay down so that we all know what we're talking about when we're speaking to each other about the parts. Okay, so there's 2 words on here that you need to know. So the concept first is that macromolecules are polymers. They're built from monomers. Mono meaning one or single. So a monomer is a single unit, or the smaller part that's going to make up the whole big thing. Poly means many. So a polymer is a bunch of monomers strung together. You can think of it kind of like maybe like a beaded necklace or something. The individual beads that make up the necklace will be considered monomers, and once you get them all on the string or whatever you're putting them on, that ending result is the polymer. Okay, so with these monomers and polymers, we know that we can start off with the single unit and we can make the bigger thing. There's also another way that reactions have to go sometimes, and that is taking something bigger and then breaking it into smaller parts. So the chemical reactions that we see come up quite often are 2 mentioned on this slide. One of them is called a dehydration reaction. So what happens is, if you have smaller parts, so let's say you have 2 monomers, or maybe you even have like a short polymer and another short polymer, and you want to join the 2 of them together, they would have to undergo a dehydration synthesis reaction. All that means is that when they come together, a water molecule will come out of the union of these 2. And that release of a water molecule is what allows the bond to be formed between these 2 molecules. So why call it a dehydration reaction? It actually makes a lot of sense why we would call it that. If someone was to tell you they were dehydrated, you would say, oh, well, they're lacking water or they're missing water. And that is what the molecules are doing. They are, in effect, getting rid of a water molecule. It's dehydrating. So the hydrolysis part is the backwards of the dehydration reaction. So hydro is dealing with water, and lysis is the splitting of, or like the cutting of. So we're talking about cutting a molecule now with the addition of water. So we get a polymer that's really long, and maybe we need to break it into the smaller parts for, recombining them to make some other molecule. Hydrolysis would be the way that we could do that. There is a protein or type or class of proteins that are called enzymes, and their job specifically is to make these reactions happen quicker. They're catalysts. They speed things up. They can be used over and over again without, being consumed. So we'll talk about enzymes more when we get to the protein portion of this chapter.
Most biological molecules are very large and are built by assembling small molecules, or monomers, into long chains. The resulting molecules are called macromolecules, or polymers. A process of linking monomers, called dehydration synthesis, involves the removal of 2 hydrogen atoms and 1 oxygen atom to form water. One way this might happen is diagrammed here, where several generic monomers are shown with OH groups that could be used for linking. The animation illustrates the joining together of monomers by dehydration synthesis, catalyzed by a polymerase enzyme, and the reverse process, in which added water results in hydrolysis, catalyzed by a hydrolase enzyme.
All right, so that was just the animation that you viewed. It's a good animation because it does show you what that dehydration synthesis and what the hydrolysis would look like. So these are just models of the thing that you just looked at. The top one is dehydration reaction, the bottom one is hydrolysis. So in the next two slides, they zoom in on each one, so I will go forward to speak on them individually. So in a dehydration reaction, you are synthesizing, or you're like making or putting together a polymer. So they give you an example where you have this short polymer here, and then you have one unlinked monomer that needs to be merged with the short polymer. And you'll notice that there's a hydroxyl group here, and then you see that there's just a singular hydrogen atom hanging out there. When these two are forced together, either with an enzyme or because they bump into each other, you can get water out of the mix, and you end up with a longer polymer that you were trying to create in the first place. So hydrolysis is the other one. So now we're going again backward. So hydro meaning water, lysis meaning splitting. So you're going to break down a polymer in this case. So here's the same exact polymer that we just created in the slide before. And when we add water to these, what you'll see is that the H2O can be split back into a proton and a hydroxyl group. And then they like cap off the short polymer and the single monomer. This is it for this lecture right here. Let's talk about the diversity of polymers very briefly. So here's the deal. You have these polymers. They're in cells. They help make up the cell. And because there's so many different ways that you can combine them, the length of them can be stretched out. Some of them are smaller. You get an immense variety of polymers being built from this small set of monomers that I'm going to introduce in the next couple of lectures. And I would like for you guys to keep thinking about yourselves, because it's kind of easy to think about yourself, eating. So when you go to eat something like a piece of bread, you guys know that carbohydrate is a primary factor in bread making. It's one of the macromolecules that you will find a lot of, especially if it's regular bread, not like a gluten-free anything, or I don't know, some other type of bread, just plain old regular bread. So in that bread, you know there's carbon atoms, you know there are hydrogen atoms, and you know that there are oxygen atoms. And if you needed any of the three of those molecules for any other reason inside of you to keep yourself going, to be sustained, all you are doing is you are breaking things apart when you chew the bread, swallow the bread, and then once it is digesting, you are further breaking it up. So you're doing a bunch of dehydration synthesis reactions along, you're doing a bunch of hydrolysis along the way, where you are taking water, you're splitting the molecules apart, and then you can store them for later use. And then if you needed an immediate, like, quick hit of energy, then you would need to go the opposite way. So not doing hydrolysis, you would need to do a dehydration reaction where water would come out and then the molecule would be split apart. This is straightforward, and we're done with this one. Put a check mark on that. I'm out.